And so is also our next speaker, Bill Regan. Bill Regan has been a perfusionist for many, many years at the Boston Children's Hospital. And he got involved with research, of course, and many product developments and also mission trips. And now he's going to talk to us about mitochondrial transplantation, or something, at least something like that. And I'm looking very much forward to this presentation because I have no idea how to do that. Thank you, Bill.
Yeah, thank you very much for inviting me and for that introduction. I appreciate it. I agree. It's great that we have a whole bunch of perfusionists actually involved in the research. It's really important that we have a piece of this too. Thank you. Yeah, and to really help out and add our expertise to some of these projects. A lot of these fellows and researchers come in and they don't have a great grasp on the perfusion component, and so it's nice to be actually involved and help. So today we're going to keep on working on this idea of Tristan. You're going to find a lot of crossover with some of the things that she had. And it's seemingly we were working on similar projects kind of at the same time. One of our spins is actually mitochondria. A couple of our docs are actually very interested in the use of mitochondria and mitochondria transplantation to help in repairing some of this tissue. So with that, I have no disclosures. Some of the head researchers, Dr. Del Nido, Dr. McCulley, and Dr. Amani, all have patents pending for isolation and usage of mitochondria. And they actually have a new consultant and company based off of that. And there will be no off-label discussion about drugs or devices. All right, so the goals of this lecture, first we're going to talk a little bit about DCD. Tristan fortunately gave us a great segue into that, so I appreciate that. So everybody's heads are already kind of in that vein, and you'll see, again, see a lot of similarities. So that's really nice. And then we'll talk a little bit about what we had to do to create a model to mimic DCD transplantation. And then we'll talk a lot about mitochondria and how that can actually help. All right, so as we all know, the organ donor pool and the amount of recipients, they don't match, right? There's a huge shortage. So one thing that has been proposed is DCD transplantation. And what that would hopefully do is open up a new chunk of donor hearts and help meet that shortage. There are a couple of problems with that. DCD organs, if you look at the schematic here, most of our organs currently are from DBD organ donation. And DBD organ donation doesn't have that ischemic time associated with it. And unfortunately, the tissues that are, the hearts that come from DCD organ donation do have an associated ischemic time that creates some injury and creates some of those hearts are unavailable to us. And so we see that they think there's the potential to increase by potentially 30 to 40% the amount of hearts available by improving DCD techniques. And that's why there's a lot of efforts put into Heart in the Box and efforts like Tristan and Stanford group are doing. So for us, we started this back in 2015. Dr. McCulley came to us and he's very interested in mitochondria. That's his first and foremost goal is mitochondria transplantation. And he had already proven through a whole bunch of papers that, and we'll look at them at the end, about mitochondria function and where it's going to fit into this puzzle. But he came to us and said, hey, I need to be able to mimic ischemic heart tissue. And so we said, I think we can handle that. So We went through a very systematic approach on how to prove each step of this to make sure that we weren't missing anything and that we could prove that mitochondrial transplantation would actually help with ischemic heart tissue. So first, let's look at mitochondria, and everybody understands mitochondria, the powerhouse of the cell, and their function in the myocardium and their muscular tissue of the heart is really crucial, especially because of the such high energy demand, the cardiac myocytes, right? And I won't read all of these. There's a lot of information here, right? ATP production, right, is probably its biggest job, right? Calcium homeostasis, and you can see there's a little blurb about each, but we will save the time for discussion, so we won't read all of that. But this keeps going on and on. The mitochondria actually quite important and do a lot of things. So reactive oxygen species, RAS production and detoxification, fatty acid and glucose, metabolism, right? And this keeps going. Apoptosis regulation, heat production, right? So we really see how mitochondria are actually extremely important in cellular function. So the first thing we needed to do is, if we're going to transplant mitochondria, we have to be able to isolate them to transplant them, right? So we had to prove a method to be able to get mitochondria to transplant. So this, when we started, was the protocol for mitochondrial isolation. And you can see there's a lot of steps, but most importantly, if you look down at the bottom, you can see that it takes a long time. It takes hours to get this and then hopefully get a good product out of it. So that just doesn't really work in the heart transplant world, right? That's way too long to do, because that's starting with once somebody receives the donor tissue, and you can't do this, say you were at a hospital, you can't start this process because then you'd have to transport it, and now you're at a different hospital. So you'd have to wait until the tissue got to your site so that you could then subsequently start this process. So one of the key factors was, can we simplify this? And we had a group of researchers, and you can see up at the top, the tissue samples that come in, about the size of a dime. But most importantly, you can see down at the bottom, this process is now down to about 30 minutes. And so this was an extremely important facet of being able to achieve mitochondrial transplantation pretty quickly. The process is also largely automated now. One of the researchers was an engineer and decided that he could make a machine that would actually automate most of this, whereas before it was a lot of pipetting and a lot of in-lab stuff. So it was really quite annoying. All right, so here, it's a very busy slide. All this says is that those mitochondria were isolated, they were mitochondria, they're purified, and they're available for use, and they actually function as they're supposed to. So these would be good, viable mitochondria for transplantation, autologous mitochondria for transplantation, right? All right, so over here on the left, you can see, on the right, you can see the damaged mitochondria, right? They seem pretty swollen, non-defined, nondescript. And the goal is to replace them over with the refined processed mitochondria. And so they actually, during ischemia, the cells actually open up and their membranes kind of open up a little bit, and they actually uptake these new mitochondria actually quite well. And mitochondria transplantation is being a therapeutic intervention that's being used quite often for all kinds of tissue now, not just in our lab for hearts. One facet of this project was to how do you get this mitochondria now into the tissue? So you've isolated it. So then there was another study that said, okay, now that we have the mitochondria, what's the best delivery method? And what we first tried was direct injection. And you can see very specific site injection at the top. But unfortunately, that was very site specific, right? So you'd have to make a whole bunch of stabs all over the place in order for this to actually work. Well, the next part of this paper, we can see that we started to go more into the root, directly into the coronary, and you can see there's definitely a lot more. The dispersion is actually quite good and covers most of the heart tissue quite easily, and it's actually pretty easy to achieve that intracoronary perfusion injection. And this is just another image of staining that shows better dispersion throughout the myocardium, and you can see the entirety of the animal there. Okay, so now we have mitochondria. We have a way to put them in. What are we going to put them in? Well, here in the lab, we wanted to prove that we were getting good results. So we need to control everything. So we had to create a model. And if you look at this model, it's horrible in a diagram, much like Tristan would say. Diagrams don't show much, but it's very similar to what their approach was. There's a collection basket. It was actually a 5-gallon water jug that was chopped in half, and the heart sat on top of you'll see in a minute, sits on top of a sheet of plastic and funnels it down to the bottom, where then it's all collected and it runs back to the reservoir. Here, I'll skip ahead so you can see this, because we don't need to talk about the schematics. That's annoying. We also have a whole bunch of random equipment sitting around. And the idea was this needs to be very reproducible. Now this was back in 2016, I think. So we dragged our S3 out of the back closet, and we started putting together pieces. And we thought the roller head was actually quite preferred, because we wanted to be able to very finely control and understand what flows we were getting. And with some of the smaller tubing, our flow probes sometimes have a little variability. So we double checked it with flow probes and with the output from the S3 itself. Okay, so here's a little bit more of a diagram showing it. This is actually split. There's an FX05 in the middle, and there's FX15s. As I'll talk about, we've run this experiment a couple of times, and some were in larger animals and some were in babies, just to prove that both tissues were improved by this. So we used stuff that was very much available to everybody. The goal for us was not to necessarily create a system that could go and transport and be used in that manner. The goal for us was to make a model to mimic the insult of ischemic tissue. So we didn't have to come up with something that could completely be transplanted, portable, but it wanted to be replicatable so that somebody else could do this similar and gets hopefully similar results. Okay, so let's switch a little bit and talk about the study itself, the DCD study, because now we have some tubing and we have some mitochondria available, but what are we going to do with it? So we wanted to mimic what would happen in a DCD protocol. So we took 40 to 50 kilo pigs. We took about 25 of them, and the reason for these was their heart size is similar to an adult at 250 to 300 grams. And we wanted to make this clinically relevant and make sure everybody understood that what we were trying to pull off. Here, I think it says it on it. Yeah. So you can see across the middle all of the things that we set up to monitor to make sure that we weren't missing anything. And then the key driver here is the red bar at the top is the warm ischemic time. And that's the biggest problem with DCD transplantation, right? That time of no perfusion when you have warm tissue happening, and that's when all of the cell ischemia and lack of perfusion happens, obviously. All right, so part of the DCD again, and this has been, the DCD stuff has been talked about in other papers, is how much time to no flow and how much time of warm ischemia is there. And we had similar results to other centers with a 22-minute average warm ischemia time. I think it's important to note that, we'll go ahead here. This is kind of mandated because you cannot touch these patients for a certain period of time, and that time is very variable across different states and countries. And so we wanted to have a very controlled number. And so if you look at our timeline, we actually did sternotomy first so that we could actually very much control, because that time is usually a little variable. And so we wanted to say, okay, no, we have 22 minutes of downtime every time. So we did some of the things first in order to control for a lot of the variables. You can see we've moved down the timeline to heart preparation, where we actually got the heart going on the system, but I'll jump ahead a little bit just to keep this going. One thing that was important in our group was that we had what we called sham hearts. So these hearts were designed to have no warm ischemic time, right? These hearts would mimic DBD donation, which is what most of us do for donation now, so that we'd have a good secondary control group. Another control group we had was just vehicle only, which was just the solution that the mitochondria in. So we'll see in a minute that we have 4 groups of hearts that we did. One is single injection of mitochondria. One is multi-series injections of mitochondria. Then we have these sham hearts to mimic what current state transplantation is. And then we have vehicle only, which is just a control to make sure that it's mitochondria, not the other solution that we're using. Right. And so this is a little bit more of the protocol here. Once we get on bypass, so you can see some of the parameters that we've been trying to control for. We also had pacing if need be. Initially, a lot of them didn't need pacing. These are to define some of the parameters and what some of the actual outcomes are. We're able to keep things pretty tight in our control groups and in our variables, so that wasn't a huge factor in the outcomes. All right, so results. So here you can see, very similar to a suspended heart over at Stanford, lying on top of a piece of plastic, right? This is immediately out of the box, out of the body, and put on the system. This is what it would look like with the injection of mitochondria. You can see off of a stopcock right there in the root. And then this is what it is not sped up. It is not, there is no extra. This was actual, would really happen. And it was really quite impressive. So we thought, all right, there must be something to this. So we kept on looking. And as you see, we start looking at some of the results here, right? And The LVDP, the DPT max, they all started coming down. One thing we won't really discuss a lot is we had two different modes. One was just intracoronary perfusion, where we just reperfused the heart. And the other one where we loaded the ventricle and we actually caused the heart to have to work at its normal rate. So we would pump 5 liters through this. And this is, our collection bucket was a little bit bigger than Stanford because it was actually trying to collect 5 liters of output and make sure that we were actually doing what the heart was supposed to do. You can see during the reperfusion, myocardial oxygen consumption, and the change here, and it all pointed towards, and this again, this is in working mode, but it's pointing towards that things are working and happening. Fractional shortening got better, right? So if you look between the two, right, these are the sham hearts again. Sham and vehicle are essentially the controls. And you can see ours had an improvement with the serial and single injections. We noticed certainly a decreased apoptosis and necrosis and good preservation of myocellular structure. We wanted to make sure that the mitochondria are actually part of this, so we did a lot of staining and made sure that the new mitochondria were the ones that were getting in there and being active. Okay, and then one thing Dr. McCulley is certainly involved quite a bit with is the cellular side of things, because he's the mitochondria guy. And so there's some quick terms over there: genomics, transcriptomics, proteomics, and metabolics. Those all help you define how things actually happened at what stage of the process. So these are some papers. The ones highlighted in red are ones Dr. McCulley worked on. And these are a series of papers that both discuss mitochondrial and enzyme activity, and then how the ischemia and reperfusion injury can cause decreased cell viability and decreased function. There's a lot of fancy stuff here. This just says, this is the metabolomics of the post-study that says, yep, these were the actual mitochondria that were actually functioning to create these metabolites. And again, there's a lot going on in this. It just says that the transcripting and the proteomics all point to this new mitochondria being the ones that are helping these cells, and they are inactive. The same, they're the predicted source of the output. But we're getting close to time, so I'll push this a little faster. So what we found is mitochondrial transplantation upregulated a lot of the processes, and those processes that it upregulated were obviously good processes. If you look here, these are all good things. And it downregulated a lot of the bad processes. So both things happened. Right? So it was really a positive in both senses. Well, so we were very happy with that. So then it was around COVID, and so things slowed down a little bit. And during that time, we decided, let's go and formalize a study, and let's do this on neonates. And you can see that we completely repeated the whole thing. And we used much smaller animals to represent the neonate or pediatric population. And those results were completely similar. All right, and I think that's a good place to stop there. And thank you.